Good morning. Thank you for joining us for Research 411, a talk show focused on sharing best practices and trending research topics delivered by professional staff and prominent UT Dallas researchers from various disciplines. I am Tiffany Willoughby, Research Education Programs and Outreach Manager in the Office of Research. I will moderate today's discussion. Joining me today are Dr. Nicole De Nisco, Assistant Professor at the University of Texas at Dallas, Biological Sciences, Dr. Jeremiah Gassensmith, Associate Professor at the University of Texas at Dallas, Chemistry, and Dr. Molly Ingersoll, Institute Pasteur in Paris. Together, they will discuss cutting-edge research related to bacteria and vaccine development. Welcome, Dr. De Nisco, Gassensmith, and Ingersoll. Dr. De Nisco, can you please share with the audience a little bit about yourself? Who you are, what you do at UT Dallas, and how it impacts our audience.
Yeah, I'd be happy to. So I am a PhD trained molecular biologist. I received my PhD at MIT, and in that environment, it was very interdisciplinary. We commonly collaborated across disciplines, and that's something that I really liked when I first was interviewing at UT Dallas. So I joined the faculty here as an assistant professor in October 2018, and since then I built a research group focused on understanding recurrent urinary tract infections, and importantly focused on trying to devise alternate therapies for them, because our current therapies for these really common infections are failing. And so this is how I met Dr. Gaston Smith when he was actually one of the first people I met outside of the Department of Biology at UT Dallas. And we really hit it off and kicked off our collaboration from there. So that's a little bit about me.
Thank you. And Dr. Gaston Smith, can you do the same? Please tell us a little bit about your role at the University of Texas at Dallas and how what you do impacts the audience.
Okay, sure. I'm an associate professor in the Department of Chemistry with a joint appointment in bioengineering, and I got a PhD from the University of Notre Dame and went off and did some work in the lab of Sir Fraser Stoddart. He won the 2016 Nobel in Chemistry, and found my way here. And so, and my background is actually physical chemistry and crystallography, and now I have merged very interdisciplinary. I've landed into a very interdisciplinary building. Actually, I came to UT Dallas to do energetic materials, applied materials research to make batteries and stuff. And I don't do any of that, and that never really actually even got off the ground. I immediately started collaborating with the people in biology and bioengineering, and so my research has nothing to do. Well, very little to do with what I trained to do, so it's been a pretty huge shift.
It sounds like you never stop learning, Dr. Gaston Smith.
No, not in this job.
Not at all. Excellent. Thank you for sharing that with us. And Dr. Ingersoll, can you please tell us a little bit about you and where you hail from? And how you came to know and interact collaboratively with Drs. De Nisco and Gassensmith, please.
Certainly, I'd be happy to share this. So I am American, but I live in Paris. Some people think that's a dream. I will tell you, there's a secret. There's a lot of paperwork here. But I trained at New York University School of Medicine. So I did all of my training in New York City. And then I had the opportunity to interview for a position at the Institute Pasteur in Paris. And it's one of the probably best known institutes for studies of infectious diseases and the way the host responds to those diseases. So I've been in Paris for almost 9 years now, where my team and I try to understand how the body responds to infection, and our focus is on the bladder. So we are trying to understand what happens when we become infected and how can we come up with these treatments that Nicole mentioned, that Dr. De Nisco mentioned, that we desperately need because our current therapies aren't working. So I got to meet Dr. De Nisco when I was invited to Texas to give a talk, and frankly, we clicked on, I think, a science level, but also on a personal level through some common interests. And when she reached out to suggest that I join this collaboration, I was really excited at the possibilities that were in front of us.
Thank you. And how long have the collaborations been underway?
Ooh, more than a year. I don't know. It seems like it's been like 12 months since March, right?
Yes.
Yeah, you know, we hit March, and then every month has been about 6 months worth of months. So
right.
So, yeah, I'm not doing too well with time right now.
That's quite all right. Well, speaking of collaboration, how in fact, Dr. Gassensmith, did you and Dr. De Nisco initially meet and begin to collaborate?
Oh yeah, so I am on one of those boards that, you know, everybody on the faculty likes, the institutional biosafety committee, and so we go through when people want to bring in things to the university from patient samples and stuff like that. We check them off, and Dr. De Nisco had a proposal, and I sat there listening to it, and I thought, well, that's interesting. And then somehow we flipped. Then I actually had to go in and I think give my own presentation, give my own pitch to the safety committee to get a protocol approved. And Dr. Nisco was there in the audience. We have a very strange board, but she was there and she was like, no, I want to stay and listen. And so I thought that was really neat. And so, you know, I had this idea to do it on bacteria. And like I said, you know, my vision of bacteria are sort of these sausages, green sausages that sit on the bottom of a dish, and you know, we use them to make proteins. I know that. Beyond what they do or why they do it, I don't know. But I figured if I'm going to do pathology of actual infection, I need an actual person who knows that kind of stuff, and it was very nice and worked out.
So you met at a safety meeting, so to speak?
Yes.
And Dr. De Nisco, if I recall, you had mentioned that you come from an interdisciplinary background. Yeah. How did the initial collaboration conversations go, and what led you to the proposals that you've submitted?
Yeah, so Dr. Gaston Smith, he approached me, and because he was developing these metal organic frameworks, which are basically things that will encapsulate particles, right, and kind of stabilize them. I guess he probably can explain it better than that. But and so he was doing this for viruses, and so he approached me because, you know, I was studying a bacterial infection, and I guess I made a good case for why it was important to study UTIs and why we need new therapies for them. And so he was like, well, could we use this as a method to generate like whole cell bacterial vaccines, right? And so that's where it started off, and I was like, well, that's a great idea, because, you know, we're kind of lacking in vaccines for UTIs, and there's reasons for this, and a lot of those reasons can possibly be circumvented by a really effective whole cell vaccine. And so that's kind of how we got started. And then I realized, you know, I am a microbiologist and a molecular biologist, and, you know, I do have clinical collaborations, and that's where we get a lot of our samples and strains, but I am not an immunologist, so we needed to bring on board an immunologist. And that's what I really love about science, is that you can just collaborate with people with all different expertises. And so I had met Dr. Ingersoll actually when I was a postdoc at UT Southwestern. I think my postdoctoral advisor invited her to give a talk, and that's how we met. And she said we kind of clicked because we both love cats and other things. And she's really like the leading expert, I mean, that I know in the field of bladder immunology. And so I thought there was no one better to have, to collaborate with. And she's just, a very amazing person. So that's why I suggested to Jeremiah that we bring her on board for some expertise. And I think it's been going pretty well.
Dr. Ingersoll, can you share a little bit more about your role in this research endeavor?
Certainly. I'm glad, Dr. Nisco, that you said it, because I wasn't sure if I should bring up our love of cats or not. Sometimes it's small things that really cement a collaboration. So essentially, when Dr. De Nisco contacted me and said, I'm involved in this very cool project, we're trying to make it's a new approach towards vaccination, but we really need the expertise of an immunologist. I said, send me the project, let's talk. And I'm going to say that it was such a simple, straightforward, elegant approach that I was immediately excited. And I think that I was 100% sold when I saw pictures of these bacteria wrapped in these metal cages, because they look to me like bacteria covered in fudge. And I just thought, this is beautiful, this is elegant. And much of our research at the Institute Pasteur is really meant to understand how we can shape the immune response, how can we make it stronger, how can we make it so that when you become infected, you know how to fight that infection better than if you weren't helped. And a vaccine is an amazing time-tested way to boost the immune response. So for me, this was perfect. And as Dr. De Nisco mentioned, we really have such different complementary approaches that it just seemed like this was kind of the perfect party to join.
Well, believe it or not, ladies and gentlemen, we do have a few questions from our audience in the queue. And the first one is, will a vaccine for UTIs be available within the next year? All right. What is the forecast? I'm seeing shaking of heads no. Do you have, some sort of a timeline the audience could entertain for things that you would be hopeful to see in the future?
I mean, the process that we're doing, maybe a decade, maybe more. The vaccines just, and this is a very critical point and very timely to mention, that vaccines have to undergo extraordinarily rigorous testing to demonstrate that they are both effective and safe. Inoculating a population with an ineffective vaccine, then they go out and start engaging in behavior that might expose them, is significantly worse than not vaccinating them at all. Absolutely. So vaccines in particular, with some of the public's concerns, just simply need to be very rigorously tested to make sure they are both safe and very effective. So this technology, we wouldn't even try to make a promise that this would deliver, but I imagine Drs. Ingersoll and De Nisco may have a bead on other technologies that are maybe moving through the clinic or research.
Certainly, I would say that I've seen some preclinical and clinical data. So preclinical meaning preliminary testing in cells or animals, and clinical data meaning testing in humans that shows some promise. I think the biggest challenge in vaccination studies is having enough people that we can say with certainty that it's safe and that it's effective. And I don't think that our community as a whole has reached that yet. And so while there are certainly promising formulations that are being tested, I think we're probably, best case scenario, 3 to 5 years away from any type of commercially available vaccine.
Thank you for sharing that information. We do have an additional question. You may have covered it at the surface level. What has prevented the creation of a vaccine? It seems like it would be somewhat common since it is a bacterial infection.
Yeah.
Well, I guess, I mean, I can try to answer part of that first. So the primary organism that causes urinary tract infections is, our favorite, one of our favorite bacteria, E. coli. And so this is something that you guys might have all heard of. But the problem is that the E. coli that cause these infections are incredibly diverse phylogenetically. And so that just means that they're expressing all different kinds of things on their surface, and they don't look like each other. So the E. coli that affects one person doesn't look like the E. coli that infects another person, and that's really important for the immune system, right? So in having when you have a lot of diversity in kind of like how the pathogens look to the immune system, then it makes it difficult for the immune system or for us to develop vaccines that kind of work against, all of the versions of that pathogen out there, right, of that E. coli. And so a lot of the vaccines that are kind of in trials right now are based upon certain what we call antigens, these immune response eliciting particles that are common to some of the E. coli, but it's taken a lot of work to kind of figure out what those are and what the best kind of antigens are to use. And these are in these subunit based kind of vaccines that are really using specific proteins and specific molecules, as opposed to our technique, which is a whole cell technique where we can just take the entire cell, and so we have all of the things that are being displayed on the surface, right, that can be used to train the immune system. So, and I don't think a lot of whole cell vaccination has really been successful with UTI vaccines, and that's because of the preparation method. And so that, I think maybe Jeremiah or Dr. Gassensmith can talk a little bit more about why current whole cell vaccination methods are not effective.
Sure. So there are vaccines against some bacterial infections. Tuberculosis is an example. It's actually not an incredibly potent vaccine either. It does an okay job in a pinch, but it's really, as far as vaccines go, kind of in the lower. It's safe, but it's not extraordinarily effective. And as Dr. Nisco pointed out, the immune system, they're just little soldiers. They're just searching for things they know to be foreign, and they identify them. And bacteria can display all kinds of stuff on their surface. They have different kinds of cell receptors, different kinds of sugars. And so E. coli being particularly good at sort of changing its coat as it goes around means that you really need to capture not just one E. coli, you need to capture many different ones. So in a patient that could have multiple different types of E. coli even that cause one infection. And so what we have done is created a method that immobilizes the bacteria in a shell. Right. So normally the way these bacteria, if you want to make a whole cell vaccine, they use something called formaldehyde, and it's not dangerous. But what it does is the formaldehyde reacts with all the proteins cross-linked. So you keep a lot of the same shapes of what's on the surface, but chemically you're changing them. OK, so they're no longer identical. They're similar, but they're no longer identical to what's on a live bacteria. Or you heat them, right? And just like when you heat an egg, it turns from a goo into a delicious morning breakfast. And so the same thing happens to bacteria, albeit not as delicious. So you physically change the proteins, and those are the only two ways that are really good at inactivating or killing bacteria, because you can't put live infections inside of people. You can try to inactivate, you can try to make mutants that are not infectious, and that's really tough. So what we wanted was a way in which we can get common, popular patient-derived samples that we know are out in the wild and inactivate them without damaging those very valuable surface, what are called epitopes, the things that the immune system responds to. And there are literally hundreds of these things on the surface of bacteria. Most of them do nothing for the immune system, but some of them certainly do, and we know that. And trying to figure out which ones do which, that's super hard. So our approach essentially just takes the whole bacteria, but doesn't disturb the surfaces, but it kills the bacteria. So you can safely inject it and release it inside the skin of a patient slowly, and that causes a very stable antibody response.
Thank you for that broad explanation. It was very thorough and helpful. Some of the audience members are curious to know what research results you have thus far with your collaborative effort.
We have a paper that is available for viewing on archive server. It is out for review at one of those journals that take a long time because they're competitive. So it's still under review, and our fingers are crossed that we'll get something back pretty soon. But it's a pretty awesome work. We can share the link with anyone that would like to have a look at the work that's under review right now.
Thank you. Another question in the queue: what are the current animal models used to study our UTI and the preclinical vaccine studies?
Really, that's our mainstay. So we use mice, and one of the reasons we use mice in general in science, we use mice because they're small, they're relatively inexpensive, and we can manipulate them genetically. But we really like using mice for urinary tract infection work because, in fact, when an animal like a mouse gets a urinary tract infection, it looks a lot like the infection we see in humans. And so what that means is the results we observe in a mouse, we can apply almost directly to humans. So we can use the same strains. So if Dr. De Nisco isolates a unique urinary tract infection strain from a patient, we can put that directly into a mouse and it will cause the same infection. They'll have the same symptoms. They'll have inflammation. They'll have the urgency and the frequency of urination. And so we really like the animal model because we think in this particular case, it's giving us even more information than we typically expect from mouse models in research. And just as an added sort of interesting side note, if you will, we don't do a lot of work outside of mice ourselves, but people also use rats sometimes. But in fact, almost every animal that I've ever investigated naturally has urinary tract infections, and this includes rodents, cats, dogs, whales, in fact. So I know I found that out. I was really excited. So in fact, what we're looking at is a naturally occurring phenomenon, and we're trying to use the information that we get from our mouse models to apply directly to human medicine.
Excellent, thank you. Dr. De Nisco, can you speak a little more to UTIs and why they seem to be so resistant to treatment?
Yeah, of course. So UTIs, there's multiple types of UTIs, and so we have to be specific. And the type of UTI that we're talking about here, that we're trying to look at these vaccines for, and that my lab is working on, is recurrent UTI. So this is a really difficult form of UTI. A lot of like UTIs that young women get, you know, you classically hear college-age women getting UTIs that's linked to sexual activity and whatnot. Those are, you know, resolved by antibiotics usually, but not always. But in the you know, the infection that I'm studying is when women are experiencing cycles of UTI. So basically, they're going to have a UTI, it'll be resolved, and they'll have a negative intervening culture, which just means that they will have a negative culture result in between, and then a couple months later, they'll have a new infection. And this cycle can happen for decades. Especially in a lot of the women that we see in the clinic. So I collaborate with Dr. Philippe Zimmern at UT Southwestern, and he's a urologist, and he has a large practice of primarily postmenopausal women. And so that's another interesting thing about this disease, is that it seems the recurrence rates seem to be higher, almost double, in postmenopausal women, and we think that this has a lot to do with hormone status, which is something that we're kind of investigating separately in my lab. But yeah, so they're really difficult to treat because they're such long-term infections that we're using antibiotics, right? And the bacteria are live, evolving organisms. So you hit them with antibiotic and you select for resistant microbes. So bacteria that are able to evolve and gain resistance are really going to then take over the population, and then you'll have a new infection with that resistant bacteria. You'll have to change antibiotics. And that cycle keeps going on until eventually you run out of antibiotics that are effective. And another important thing that happens, especially in elderly women, is that patients develop allergy to the antibiotics that they're taking. If they take it for a long time, which happens a lot with these women, is they'll be taking antibiotics not just for like 7 to 10 days, but for months. They will develop allergy to them and no longer be able to take them either. So they just run out of treatment options because we aren't developing antibiotics quickly enough. I think it's more of a issue with pharmaceutical company development than with ideas from us researchers, but that's a whole different story.
And yeah. What happens if a UTI goes untreated? What kind of risk does a patient have?
So UTIs can actually develop into, a bladder infection can ascend up into the kidneys, and if left untreated there, it can actually go into the bloodstream and cause sepsis, and which can be fatal. So actually, I suffered from this when I was in high school. I almost died from a UTI that had ascended into my kidneys, and the physician was not very experienced and gave me a shot of Cipro directly into my kidneys, and that basically killed the bacteria. Were already in my bloodstream, and it killed them all, and I almost went into toxic shock, and yeah, so. And this was, I was a healthy young individual and not had any of the risk factors for this infection at all, but besides being stressed out high schooler. And so that's really interesting that I never planned to work on UTI. It just kind of happened, but it just shows like how common this infection is, and that it's something that women deal with like as part of their lives, and men, of course, when you're talking about catheterization and stuff like that we just don't talk about as much as we should as a society. So it can be bad. I know from experience.
Absolutely. Thank you for sharing that.
Yeah, of course.
Now, tell me about how our university, the University of Texas at Dallas, and/or the Office of Research have been able to support you with your research and/or collaborative research efforts.
We are very fortunate. We have a very forward-thinking research enterprise here that established small amounts of money to fund very collaborative projects, and this is such a clever funding mechanism. It really is. And so what you apply to university, it undergoes peer review at your university, and you have to find somebody not in your area, right? So this was a fantastic opportunity to do a lot of this research, because vaccine research is unbelievably expensive. Every time you bring animals in, it costs a lot of money to get these things going. And animals, the husbandry, the taking care of them, they're fed, they're taken well care of. Every day there's a fee associated with making sure that they're healthy and reasonably happy down there. So UTD has this funding mechanism for joint collaborations that across departments called SPIRE, and Dr. De Nisco and I wrote one, and we were fortunate that our colleagues thought it was competitive, and that sort of cemented it, because now not only did we have a mutual interest in this project, but we also now had some resources to really get good preliminary data, and I think we have some really great preliminary data. It helped bring Dr. Ingersoll into the fold, and and now we have an international collaboration with immunology and microbiology rock stars, and we can really, I think, do something really great. I think we've got a lot of next generation plans, and I think this is quite exciting. Yeah.
Thank you very much for sharing that information. Can you all provide advice to graduate students interested in pursuing collaborative research projects? Because you've all been there before, correct? You were the graduate student at one time. Is there any knowledge you could share with them at this stage in your careers? Anything to help spur them along as they continue with their research endeavors?
Sure.
Go first.
So I would say, if you are pursuing a career in science, you are going to always be learning. So never feel like you are done learning, and I think just being curious about other fields, and just taking the time to like listen to something that's outside of your field. You never know, I mean, where that can take you, and I really feel like that's where innovation really comes from, is when different fields meet each other and collide. So don't be afraid of something if it seems unfamiliar to you. And if you're listening to a talk and you don't understand something, or if you're talking to a potential collaborator and you don't understand something, ask them, tell them that you don't understand, because they're people, they're nice people most of the time, that are going to be happy to explain it to you. And just being open and being humble, and in knowing that your knowledge is not complete and probably will never be complete, because we are all constantly learning. And yeah, I that's how I feel. And finding a mentor that's really open to collaboration is important too, because of course your mentor has to support that collaboration. So so finding someone that is just kind of intellectually curious and wants to learn about different fields. Do you have any other advice?
I would add on very specifically, I think you're absolutely right. I think what I would add on that I wish that I had done more as a PhD student is really specifically talking to people, really going up to people and saying, hey, I have an idea. What do you think? And the reason I'm talking to you is because I think we can work together in this way. And I think I spent too much time waiting for the introduction, and you can't. You have to jump in there, and Dr. De Nisco's absolutely correct. We're all essentially here because we're curious. So don't be afraid of the big professor. Don't be afraid of the senior postdoc. Don't be afraid of another grad student. Go and give them your ideas and open a discussion. Don't wait.
Perfect. That's excellent advice. I do have another question for you. It's most specific. Do you currently have a need for any additional research personnel?
Who
Yes.
wants to go to Paris?
For us, come check out our website if you think the research is interesting. We are looking to hire right now at any and all levels. So if you think what we do is interesting and you're interested in living in Paris, when the borders open up, we can talk.
Excellent, Dr. Ingersoll. If I could ask Dr. De Nisco and Gassensmith to maybe share any last minute thoughts or considerations with our audience today.
I'll let Jeremiah go first because he has to skedaddle.
Right. I think that last comment by Dr. Ingersoll was really on point. If you're curious, be curious. Just ask questions, follow whatever you want. I've now been teaching undergraduates and graduate students for 7 years, in addition to having an ungodly amount of time in higher education as a student. I've never seen anyone die from asking a question, much less being wrong. Maybe slightly embarrassed, but that wears off. If that's the worst thing you could possibly expect, is to be modestly embarrassed, or if some jerk gives you a hard time because they're a jerk, it's them, not you. So just be curious, follow what you want to do, and don't let anything stop you. And yeah, that I think is what I would strongly recommend to anybody listening.
Thank you very much. So perseverance, tenacity, great words to use to encourage everyone to keep being curious. Well, thank you very much, Drs. De Nisco, Gassensmith, and Ingersoll, for being on our program today to share valuable information with our community. Registration and so much more can be found at research.utdallas.edu. Thank you.